Okay, 6 says that scientists can quantify the rate of translation as ribosomes move along an mRNA from one codon to the next using a procedure called ribosome profiling. The scientists measured how long a ribosome remains stationary at each codon of each mRNA. They determined the average translation rate across all codons is 5.2 amino acids per second, but that the average translation rate of specific codons in different mRNAs can vary widely. These variations in translation rates are thought to facilitate correct folding of the protein being produced. The rate at which 3 different codons were translated was measured in 100 different mRNAs. The scientists determined the distribution of rate, so the number of times each rate was recorded. For each of the 3 codons, you have GAC, AUU, and UGG. Okay, so these graphs are a little bit different from what you're used to. They're not the standard bar graphs that we made in, I believe, question 2. These are more so what are known as histograms. And so what histograms are they're really just the count of how many times a certain instance occurred. So the amount of times that they recorded, 150 milliseconds per codon was 20. So they got 20 times, so they got that rate 20 times. So this is more so a histogram for count. And the important thing about this one is to make sure you're looking at the units, because I had some students read these graphs kind of the opposite way that you should. But if you just paid attention to the units that they gave you, it would indicate, what you're supposed to be looking at. So you've got these numbers down here on your x-axis, and it's in milliseconds per codon. So this would be 150 milliseconds per codon, and then over here would be 650 milliseconds per codon. So over towards this end is actually the faster, and then towards this end is slower. Okay, because this indicates that it takes less time to translate one codon. I had some students, if you read this incorrectly and thought it was like codons like per second, which I believe they kind of reference they reference that in the question. However, you have to be mindful of what they actually give you in the graph, which is milliseconds per codon. However, if this was 650 codons per second, then it would be opposite, where the opposite side is considered faster. But since it's milliseconds per codon, everything closer to the left end of the graph is going to be faster, and then everything to the right end is going to be slower. Okay, so if you were confused by this one, just keep in mind what the units actually are and what the bottom x-axis is indicating. Okay? So let's go into our first question. It says, using data in figure 1, graph A, identify the rate in milliseconds per codon that was recorded the greatest number of times for the GAC codon. So figure 1, graph A, we're looking for the most recorded times. So this is this graph over here. If you zoom this in, you're looking for the rate that they want you to report it in milliseconds per codon. So you're looking at the x-axis. Which one of these was recorded the most amount of times? Okay, recorded the greatest number of times for GAC, and our top graph is GAC here. So we're really just looking for the highest number that was recorded. The highest one in here is this bar right here. If you look at what the rate is, it's 150 milliseconds per codon. So the answer for that one is very straightforward. You would just put 150 milliseconds per codon and make sure to put your units. Okay. And then part B says, using the data in figure 1, graphs B and C, describe the variation in translation rate of the AUU codon compared to the UGG codon. So we're looking and comparing B and C. Okay, so this is where it's important that we kept in mind which side of the graph is, the faster rate and which one is the slower rate. So if we look at B, you have majority of the recorded events happening, between 50 and 200 milliseconds per codon, which is relatively fast. And then if you look at UGG, it's much more spread out, and you have it ranging anywhere from like 50 to 950 milliseconds per codon. But on average, this one, it's more towards the other end if you were to average it out. Now, for this question here, they're asking you to describe the variation. Now, for this one, Variation usually is going to indicate, in my opinion, the spread of the graphs, because they do have very different types of spread. For this one, I would probably mention spread as well as on average, which one is faster, just in case. So I would say that AUU, so our graph B, is less spread out. Or you can say less variation, more specific is another way you can put it. And then you can say compared to the other one, compared to a UGG, on average, and I would mention on average, because you do have obviously a spread and you have multiple numbers, on average, faster translation rate. Now for UGG, it would just be the opposite. Okay, so more variation, because we saw a really big spread of that data, it probably would have a very large, like if we're thinking about it in the fact of like significant error, and then on average, slower. Okay, so that would be B. You're really just reporting the values that you're getting. C is scientists hypothesize that tRNA molecules that bind to UGG codons are available in lower abundance than tRNAs that bind to AUU codons. Support the scientists' hypothesis using the data in figure 1. Okay, so this is why I think B is more so referring to spread, because C is referring more so to rate. So, which one's faster and which one's slower. They're saying that the UGG codons are available in lower abundance. That tRNAs that code for them are in lower abundance. We know that the tRNAs have to bring the amino acids over to the codon to obviously do translation. So if there are less tRNAs available, it's going to slow down translation, right? It's like doing a project with 5 people versus doing a project with 500 people. If you basically have less workers, it's going to take a lot longer. So that does make sense with our data, because when we looked at UGG, we said on average, the rate is much slower because you're getting most of the bars around this region here. I would say maybe on average about 350 is where that middle part is. Whereas here you're getting the highest amount around 100. So it's very clear that this one is going a lot slower. Like we indicated in part B, I would maybe re-mention it. So it does make sense with figure 1 because UGG has slower average translation rate. And so since the translation rate is slower, it's likely that there are less tRNAs, likely less tRNAs specific to UGG, limiting rate. Okay. So you really just had to justify the fact that in figure 1, you can see that UGG is slower, and UGG being slower does make sense if it does have a lower abundance of tRNAs, right? It's going to take longer to find the right tRNA and bring it over, which is going to slow down translation rate. Now the last one, D, says amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently than in mRNA than do other codons. Based on the data provided, explain why the use of one codon over another for the same amino acid might result in increased level of protein production for a particular mRNA. Okay, so this really just goes along with C. I'll put it right here. We'll put D here. Explain why the use of one codon over another for the same amino acid would result in increased levels of protein production. Okay, so I would say that for this one, we can clearly see that just based on these 3 graphs, that codons have different rates of translation. Okay, so codons tend to have different rates of translation. And to justify that, or to give an explanation why, I would say because, they're limited by how many tRNAs they have. So codons have different rate of translation because of tRNA availability. Slower translation. So they don't actually, you have to make the connection between rate of translation and protein levels. So you have to say that slower translation would result in decreased protein levels, okay? So since codons are different in how fast they translate, they're also going to be different in how much protein they're able to produce in a given amount of time. So for example, UGG, our slow one over here, is not going to be able to produce as much protein in a given amount of time when compared to AUU, for example. So the protein expression is going to be a lot lower in UGG just because it takes longer to make the protein. So slower translation means decreased protein levels. So you have to make that connection. I feel like that's where you'll get the point. Decreased protein production. Okay, so as long as you connected those two, I think you should get the mark for that one. Okay, so that's actually the end of our FRQs. We did all 6 of them. If you have any questions, please let me know in terms of, if you had different answers for these and you want me to check them, or if you're wondering what counts and what wouldn't. We will have to wait for the actual marking scheme to come out so that I can give you guys 100% answers for these, but this, in my opinion, are the correct answers for your FRQ questions for this past AP exam. Okay, thanks guys.